Hello everyone. My name is Merta Ramzani, and I'm a PhD student in Dr. Kuzum's group. Today I'm going to give an overview of our group's research, which is based on multimodal experiments and multimodal neural data analysis. So we know that brain is one of the most sophisticated systems that comprises billions of neurons that are connected through intricate and complex neural networks to process sensory information and allow us to perceive the outer world. When we open up this complex system, we find dynamics and connections with a broad range of spatial and temporal scales. For example, the neural circuits are at centimeter or millimeter scales, and the building blocks of these neural circuits, which are neurons, are in the scale of micrometer. And the ion gates and also proteins that makes these ion channels are in the range of submicron and also nanometer. In addition to such diverse spatial scales, we also know that the neural dynamics span a wide range of temporal scales from sub-millisecond for action potentials all the way to days and hours and days for learning and memory. So over the last decades, lots of different methods and techniques have been developed to capture and monitor these diverse neural activities. Electrophysiology is one of the major techniques that have been widely used in neuroscience to record electrical signals generated by neural populations. Optical imaging and genetic modifications developed in early 2000s also enabled neuroscientists to investigate neural circuits at single cell resolutions. So the urge to record from more and more number of neurons have pushed the field to constantly improve. And as you can see, for example, in this figure, the number of neurons that have been recorded using electrical or optical methods have been exponentially grown during the past years. And as mentioned by Dr. Kuzum, it resembles the Moore's law that we have in semiconductor industry. So although electrical and optical methods are widely used to study the brain, they have their specific challenges. For example, electrophysiology, although it provides very high temporal resolution, when we want to record from, for example, let's say thousands of neurons simultaneously, we need to implant lots of electrodes inside the brain. And it will obviously damage the tissue and also limit the behavior of the animal models or even humans. And even though that these probes also record from single neurons, they are very limited to just neurons that are in close proximity of the electrodes, maybe in 100 micron or 200 micron at most. And on the other hand, optical imaging methods have enabled us to record the activity of large populations of neurons, even up to 1 million neurons, although it is at very low frequency sampling rate. But they require very expensive and bulky microscope setups, which limits the animal's behavior. And unlike electrophysiology, optical recording technologies provide greater spatial resolution. We can image the subcompartments of neurons up to dendrites, but the temporal resolution of optical imaging techniques is very limited due to the slow kinetics of indicators, for example, the voltage indicators or calcium indicators, and also the low frame acquisition rate of microscopy setups. So at the end, none of these technologies can capture neural dynamics with both high spatial and high temporal resolution. So to address this challenge, we follow a different approach where we combine high temporal resolution of electrophysiology with spatial resolution and cell selectivity and specificity of optical imaging in multimodal experiments. To integrate electrophysiology with optical imaging, however, we need to have proper neural probes that can be simultaneously used with optical imaging setups. So in recent years, different types of transparent and flexible materials have been used to develop such neural recording devices that are compatible with multimodal experiments and imaging setups. For example, polymers such as Perlin C, SU8, polyamide PDMS, and the list goes on, have been utilized to form the substrate and also encapsulation of these neural probes. And metals are mostly commonly used for the electrodes and interconnects of penetrating probes that go inside the tissue. But the range of materials that have been used for surface and planar electrodes have been very different, all the way from metals to carbon, graphene, transparent conductive oxides, and the list goes on. And as you can see here in this table, although it might be very small and hard to read, so there have been several multimodal experiments using different types of electrodes fabricated using different range of materials in the recent years. And in this talk today, I will review some of the multimodal experiments that we have done in our lab during the past years. So we have tried to employ different modalities to record local, in some examples, we have recorded local brain activity using our transparent graphene electrodes that are implanted over the surface of the cortex. And at the same time, we try to perform imaging using different types of microscopy. For example, in this case, we use wide field calcium imaging to capture the neural dynamics across the entire surface of the cortex in mice. And in these experiments, our goal was to investigate the possibility of predicting the global activity across the entire brain from just some local recordings using the graphene electrodes. In another type of experiments, we took advantage of the optical recordings to capture neural dynamics from different layers of the brain. And simultaneously, we recorded neural signals at the surface of the brain. So by using multimodal processing pipelines, we tried to predict neural activity at deep layers of the brain just by using the surface recordings. These models could potentially transform the BCI field by providing inference pipelines that can be used to predict the activity of neurons in deep layers of the brain, let's say layer 5 in motor cortex, by only using the surface potentials recorded by micro-ECOG arrays from the surface, which are significantly less invasive compared to penetrating electrodes. So I will now explain 2 examples of these studies that we have done in recent years. In this specific study, we implanted 16-channel planar graphene arrays over the cortex of mice to record local electrical signals. And at the same time, we performed wide field calcium imaging across the entire cortex. And we used this multimodal dataset to train a simple neural network model to predict brain activities across the entire cortex by only using the signals recorded by the 16-channel graphene electrodes. So we use the power of different frequency bands from delta all the way to high gamma of all of these 16 channels as inputs to our model. And the goal was to predict the average activity of a specific brain regions, for example, visual cortex or motor cortex, or the components that we extracted from raw images using ICA decomposition. So our decoding results on the bottom, I can play this one. Yeah. On the left side, you can see the predicted and true average calcium activity for different areas of the brain, for the contralateral and ipsilateral sides of the brain. And on the right side, you can see both the grand truths and also predicted calcium fluorescence from which are predicted from the recordings of the 16-channel graphene electrodes. So in a more recent study, we implanted an updated version of our graphene electrodes, which has 64 channels. And the electrodes on these arrays are scaled to 20 micron in diameter, and previously was 100 micron, which I will later explain how they helped us to get better signal quality. So the goal of this experiment was investigation of the possibility to predict neural activity deep inside the brain by only recording the surface potentials. So we used the multimodal datasets to train a model similar to previous work to first predict the average neural activity in the field of view for both layer 1 and layer 2 tree of the visual cortex. Unlike the previous work, here the small size of the electrodes enabled us to record high frequency activity with higher reliability, and specifically the MUA band, which stands for multi-unit activity, and it is highly correlated with the spiking activity of neurons inside the brain. So with having these different frequency bands from low frequency all the way to very high frequency, we could successfully predict the average calcium activity in layer 1 and in layer 2 tree just by using the power bands at different of all of these 64 channels. So having the first goal of our study achieved, we took one step forward to see if we can predict the activity of individual neurons that we imaged in layer 2 tree. So to do so, one solution was to just scale the previous model and trying to predict the calcium activity of individual neurons by increasing the number of output layers. But from neuroscience, we know that most of the neural activity that we record from, for example, 136 neurons in layer 2 tree in this specific experiment, they are not actually in 136 dimension. So we know that most of these neurons are highly connected to each other, and it helps to ensure reliability, and also it helps to significantly decrease the energy consumption of the brain. So we decided to use dimensionality reduction techniques to find a low dimensional space that fully represent the neural activity captured by 2-photon imaging. So by using this specific types of neural dimensionality reduction technique, which is called GPFA, and it was originally developed for spiking activities, but here we just applied it for calcium activities. And we identified 8 latent variables that almost explain 99.9% of the variance in the 2-photon calcium data. And using these 8 different latent variables that we identified in the 2-photon imaging results, we used them. We tried to use the powers at different frequency bands from the ECOG recordings to predict each of these different latent variables. And finally, we just projected the inferred latent variables or predicted latent variables to single cell dimension to reconstruct the calcium activity of individual cells in layer 2 tree. So our decoding results, as you can see here for 5 example neurons, we could successfully decode the activity of some of these neurons, not all of them, and we are still working on it to improve the dimensionality reduction and also the prediction model to further improve our technique to predict the activity of more neurons in layer 2 tree. And here in this figure, you can also see the correlation between the predicted and true calcium activity for all neurons that we imaged in layer 2 tree with their location identified in the field of view of 2-photon imaging. So at the end, I would like to acknowledge the funding agencies, collaborators, and group members whose help was crucial for these experiments and studies. Thank you.